Okay, I know some of you have an upcoming midterm in Bio 220 this week, so I'll try to keep it relatively short. Also, just to give you some feedback on the midterm, the TA should be done marking the short answer questions today, and then we'll be putting together the marks. We're hoping to have them uploaded for you sometime by Wednesday morning or early afternoon. I think most of you did fairly well based on the short answers that we were taking a look at, and I'm hoping that it's in line with what you're hoping that your performance was like on the midterm as well. So I'm going to switch gears just a little bit, and I'm going to talk to you a little bit about what you've been doing in your tutorials over the last day or so. And one of the things that is really interesting about science is that science itself is controversial, and science is controversial for a number of reasons. And what you've started looking at today in your tutorials was really one of those big controversies. In fact, we're going to look at two of the biggest controversies right now in biology, and it deals with some of the biggest names in research and whether or not these proteins, one of which you've looked at today, irisin, actually exists or not. And it's surprising that there is so much controversy around a gene and a protein product, and this makes it very, very difficult for us as biologists, as students, and as profs to really make sense as to what is going on in the research. And this will lead into the module after exercise dealing with what is our standard of practice in research, and how do we actually make sure that these publications actually mean anything. The other thing that I wanted you to think a little bit about, you've heard a lot about exercise being good for you, but I want you to think a little bit about what exercise actually entails. Why is exercise good for you? Is exercise simply good for you because people say that it's good for you? And then secondly, how often do you actually have to exercise? If exercise really is a magic panacea that will fix everything that I'm going to be telling you about, how often should you do it? And then who is right, who is wrong? And one of the reasons why this is such a controversial field right now, and this is the paper, I think, that you guys were looking at, not this paper, but the paper after this. This came out, by the way, 5 months before the paper that you were reading. So 5 months before the paper you were reading, everyone in the scientific community was coming out strongly against irisin. Irisin doesn't exist. So imagine you went around and you told your parents that you have a 4.0 GPA. In your 4.0 GPA, your parents ask you, show me the proof that you actually have a 4.0 GPA. And let's say that you actually have the proof, but no one believes you. It's like, oh, you must have hacked into the computer system, or you must have done something to make yourself look like you have a 4.0 GPA. And then what are you going to do? Have your parents come over and talk to each of your profs individually? And this is sort of where we're at. So irisin, this molecule, this magic molecule induced by exercise, was originally described just a few months ago, or 1 year ago, as being a complete myth. Now imagine that's your body of work, that someone is saying that everything that you're doing is a myth. It doesn't exist. So again, I don't know how much science background you have. This is that paper that you were reading, the one that was published in Cell Metabolism. But this came out only 5 months later. So again, think about how quickly these experiments must have been put together, and how quickly these authors responded to this detracting statement that everything is a myth. And again, the titles of everything that came out was, irisin is not a myth. It is not something fictitious. It's not a unicorn. It actually does exist. And we're going to show you that irisin actually does exist, because people's reputations are on the line. Bruce Spiegelman, who is the senior author at Harvard, is a renowned endocrinologist, and he's the senior author on that paper. And he had to come out very quickly and very forcefully to show that, in fact, irisin was not a myth. So we're going to eventually get to that paper, the one that you've been looking at in tutorial today, and I'm going to take you through it in some more detail. That paper is still controversial, by the way, even though he came out with that paper in August of 2015, a lot of people still don't believe that that paper is true. And we'll go through some of those different ideas as well. But I want us to look at when we look at papers, how we should be looking at papers and whether or not these controversies are real controversies or controversies that can be accounted for in different ways. I also want you to get an idea when you hear all this stuff about exercise being good for you. I want you to think, like, why is exercise good for you? You know, if you have a chronic headache and you take Tylenol for your headache, you know that the effects of Tylenol wear off over time. And do the effects of exercise wear off over time? And these are some of the things that we'll be talking about. Some of the anti-inflammatory effects of exercise, how exercise releases stress hormones like cortisol and adrenaline. And all of you have heard that exercise is a good way to combat stress. We'll be talking about another classic molecule, interleukin-6 or IL-6 from working skeletal muscle. This is where that whole idea that something from working muscle is secreted. This is where the grandfather to irisin came about. And then we'll talk about some of the other cells that are involved, like macrophages as well as monocytes. So why are we so interested in exercise? Oh, and I have to apologize. There's no really good segue to get into this other than, do any of you want any ice cream? Two caveats. You can't be lactose intolerant because that would be really messy. And then secondly, you have to kind of want to eat ice cream. I'm not sure if it's melted or not, but if you can help yourselves, it's mainly because a student group was selling this. I have chocolate, which is good for you, and then strawberry. If anyone's interested, yep. Okay. So why don't you guys go that way? And let me talk to you while you're eating and maybe think a little bit about exercise. There was a paper, by the way, that showed that you have to do nothing more than actually think about exercise to actually benefit from exercise. Do you think that paper's correct? Like, all you have to do is think about exercising, imagining that you're exercising, and then you get all the benefits from exercise. Good paper? No? Something intrinsically makes you want to think that it's wrong, but in fact, the science behind it seems to be fairly good. So there is this increase in obesity that we're well aware of from the first part of the course, and we know that there are a lot of other conditions that arise. We know that metabolic syndrome leads to type 3 diabetes, which lead to other forms of diabetes. We know that there are cardiovascular risks. We know that there's an increased risk of things like cancer. We know it affects your breathing. You will develop chronic pulmonary obstructive disorders, and it may affect your mental health. So we know that having this overall change in obesity, and as we become more obese as a population, actually leads to a lot of other diseases. There's a lot of evidence to suggest that that is correct. We know that as you start to become obese, some of your adipocytes, and just for clarity, I've actually changed this in your notes to interleukin-1 from interleukin-6. We're going to come back to interleukin-6 several times, and I'm going to point out a couple of differences for you. But we know that as you start to become obese, that you start to increase the levels of circulating inflammatory markers. You may not have an underlying infection, but your body is responding to you being obese just like you have an infection going on in your body. So you'll have increased levels of circulating interleukin-1, tumor necrosis factor, C-reactive protein, all markers, classic markers in pathology for you having inflammation. But no underlying bacterial infection or anything else. And we know that this accumulation of visceral fat leads to the accompaniment by pro-inflammatory immune cells. Those immune cells also secrete different types of things. We know that the adipocytes release different types of adipokines that are pro-inflammatory, and it becomes very unhealthy. And we know that one possibility is that exercise may simply work in the reverse way, that exercise is anti-inflammatory, that somehow, for using mechanisms that we're not 100% sure of, that exercise might be anti-inflammatory and as a result might be beneficial. Now there's absolutely no doubt. There's no doubt in the literature whatsoever that exercise, when done appropriately, is actually very, very beneficial to you. There's no doubt, for example, that regular exercise will promote cardiovascular health. That has been known for over 40 years. So no doubt about that whatsoever. We know that certain forms of exercise will help to increase your the good blood profile. So you'll lower some of the bad triglycerides, you'll raise some of the other high density lipoprotein containing particles or the protective HDL cholesterol levels. And we know that exercise has all of these beneficial effects. And we also think that the exercise also has some very beneficial effects against chronic inflammatory associated diseases as well. And there are a number of these related to like rheumatoid arthritis, etc. So we know exercise is very, very beneficial. But as biologists, we have to know the mechanism that underlies exercise being beneficial. So is it simply the fact that it lowers, somehow it changes your inflammatory status? It might be. That might be one possibility for how this works. So if that is actually the case, then we have to go back to your adipose tissue. And just when you thought that you could forget all about this, that the final exam isn't cumulative, that all that stuff that you had shoved into your brain about type 1 and type 2 macrophages, you could let it all go, bring it all back, because you're going to need it for your final exam. But I'm hoping it'll make some sense to you as well. And we had talked about having a healthy diet, having normal physical levels of activity, and then having certain types of macrophages, M2 macrophages, CD4 positive T cells, or your immune cells in this particular instance, and that if you start becoming obese, your immune cells start changing. They go into the CD8 positive type of T lymphocytes, and we know that you have changes to the M1 type of macrophage, and we have an infiltration, a removal of one type, and the gain of the other type. And we know that when these types of things happen, we start producing more of these pro-inflammatory cytokines as we increase the size of these adipocytes, that we have dysregulation of our lipids. We have the bad lipids, the LDL type of lipids, increased triglycerides in your blood, increased free fatty acids. The different cell types will all increase their toll-like receptor expression systems. And we know that it's been accompanied by a number of other health factors as well. Things like type 2 diabetes, increased risk of chronic pulmonary diseases, etc. So we know that overall, that this system is an important system, and it's not just the accumulation of fat, and it's not just the release of pro-inflammatory cytokines, and maybe exercise has some impact on these different adipocytes, but I'm going to also suggest to you that it's not the only mechanism, and we'll take a look at some of these other ones as we go along. So again, exercise has a number of anti-inflammatory effects, absolutely true, but we know that it goes beyond just anti-inflammation, that there are other effects as well. Let me just ask you before I get into this a little bit later on, how much exercise do you need per day? So let's say I want to stay nice and healthy. Let's say I want to have adipocytes and my metabolic status over there on the left. How much do I have to exercise each and every day? Do I have to exercise every day? Do I have to eat vitamins every day? What should I be doing to maintain my adipocytes like these here on the left? What have you heard in the news? Yes. Okay, 150 minutes of exercise per week, about 30 minutes a day, 20 to 30 minutes a day. But what does that mean? So, for example, I consider eating a form of exercise. So should I be eating for like 30 minutes a day? Like what is exercise? Sorry? Okay, so I can tell you that when I see a hamburger, my heart rate goes up like really, really high. It's like I get very excited, I get out of breath, I have like, it looks like I'm exercising. I even start sweating, you know, all that, you know, so I'm not sure. So we'll talk a little bit about this a little bit later on, but I think all of you qualitatively know that exercise does involve an elevation of your blood pressure, elevation of your heart rate, and that you should be at least breaking out into a modest sweat. So we'll kind of use that as a guideline, and you are 100% correct. About 150 minutes per week, or about 20 to 30 minutes a day. And most people use that as sort of a form of brisk walking. Now, is that really all that beneficial to you? So I'm just going to point this out. You don't have to memorize everything that's on this diagram. So when we talk about cardiovascular diseases or coronary heart diseases or CHD that you see here, when we talk about stroke, when we talk about things like cancer, when we talk about type 2 diabetes or dementia, other factors like mental health down here at the bottom, I need you to understand that exercise has been shown to be beneficial in all of these. When we exercise, we reduce the likelihood that we're going to have coronary heart disease. Now, that comes with a caveat. When you take a look at all of the different disorders and diseases that have been looked at by a number of researchers over the last 40 years in this really nice summary diagram, there are two different types of studies that are actually indicated for you here. And you don't have to worry about reading through. If you do, that's great. But as you're looking for papers for presentation, a lot of these studies are observational. Like if you exercise, you do better. If you exercise, you don't have to stay in the hospital as long. Now, those are really powerful types of studies. All of these, if you take a look, for example, with dementia, it's an observational epidemiological study. Okay, there is no intervention being done, it's just an observation. Some of these involve things like RCTs or randomized control trials. They're broken up into control groups and groups that are actually being experimented on. And these types of studies, again, you don't have to worry about the details, are ones that I want you to be thinking about when you're doing your presentation, when you're hearing about different things, when you're hearing about Bruce Spiegelman's work with iRisen. Are you able to at least identify if this is observational, if this is a randomized control, or if this is an experimental type of paper that you are reading through? So is exercise beneficial? The literature is overwhelmingly positive that yes, modest or moderate exercise is incredibly beneficial to every single condition that we can look at. But again, we're biologists. Just knowing that something is good for you, maybe your Haagen-Dazs ice cream is good for you, maybe Twix bars are good for you. I like to think that Twix bars are incredibly healthy for you. But whatever it is, you have to be able to assign a biology to this. So this is what we're going to try to do over the next week or so. So is exercise actually a form of medicine? Because in theory, one of the reasons why there's so much debate about iRisen is if I can isolate the pathway that turns on iRisen, I actually don't have to exercise. All I have to do is bump up iRisen in my blood by taking a pill, and then that's enough. All the benefits that I get from iRisen is enough. But if I'm the first person to show that iRisen does all those things, then I'll be rich. But in fact, exercise as a form of medicine, taking it as a pill, is probably not the way in which we think of exercise. So the effect of exercise may be just simply changing our fat status, changing the visceral mass of our adipocytes. It might be changing the pro-inflammatory cytokines, reducing those, and it might be increasing anti-inflammatory cytokines. But keep in mind one thing. When you take a pill as a form of prevention or because you're sick, you have to keep taking that pill, and you have to keep exercising in order to get these benefits. That means, on a fundamental level, that there is a mechanism that's involved. If it just stayed high, I only exercised once in my entire life, and I reap the benefits of having exercised my entire life, it means that there isn't something that is unique to exercise, that it just happens by magic. And we know that exercise has to be repeated in order to get these proper effects, that every bout of exercise leads to this improvement. But we need to understand what are the mechanisms that are involved in this process, and we have to also look at whether or not exercise really is preventative. So it's one thing to say that exercise can help with cardiovascular disease, but if you don't want the disease in the first place, can exercising before you get the disease, before you develop the disorder, actually help? Because that's the hope for people that might have dementia, that if you start exercising now, or your parents start exercising in their 50s, that by the time they reach their 60s, it might prevent them from developing dementia. So maybe exercise can be either preventative or it can be a way to treat different types of diseases. So again, the simplest way that exercise might work is to simply reduce visceral fat mass, reduce the adipocytes, and as a result, exercise might, by changing the visceral fat mass, change the anti-inflammatory cytokines that are being released from those fat cells. It might also cause the increased production of different myokines from contracting skeletal muscle. And we call these myokines, as you've learned in your lecture or in your tutorial today. Or it might be because we are affecting these toll-like receptors that we looked at for inflammation in other systems. So all of these things together might actually lead to the production of or a change in pro-inflammatory cytokines. It might alter these major histocompatibility complexes, and as a result, it may change your or your inflammatory status. So these are some important slides coming up. Based on your feedback, and I appreciate you guys taking the time to give me feedback, we're going to keep most of how we do things the same. But at the end of each lecture, instead of writing a whole summary for you on the announcements page, at the end of each lecture, I'm going to tell you which types of things that you should concentrate on from that particular lecture. And some of the slides that are mentioned specifically for you at the very end. We have these two different cell types that infiltrate and get into adipose tissue. And these are these monocytes as well as macrophages. We know that as you go from being healthy to being obese or to being overweight, that the types of monocytes and the types of macrophages change. We call this phenotypic switching. They go from the M2 to the M1, and we know that we go from CD4 to CD8 positive types of cells as well. And this phenotypic switching within adipose tissue seems to be really important. We haven't really talked about why this occurs and even how this occurs. We know that if you take a look at the peripheral blood after someone has exercised, that there is a change in the number of different types of cells. We know that there is a decrease in the pro-inflammatory monocytes after someone has been exercising, and we also know that there is an increase in the T regulatory cells after someone has exercised. So again, this is not simply observational. You can go in and take a look, see what types of cells are being affected, and you can almost imagine the types of experiments that you would want to do following exercise. So exercise changes the types of cells that are normally found. And these changes in those pro-inflammatory monocytes and the increase in circulating Treg cells, and I'll ask you to keep in mind these Treg cells. You'll understand why these Treg cells are important in a few slides, why these mechanisms might be important for anti-inflammation following exercise. Again, we know that we have type 2 diabetes, cardiovascular disease, dementia, several cancers, breast cancer, for example, ovarian cancer, and other types of cancers with exercise have been shown to be impacted. If you exercise, cardiovascular disease risk goes down. If you exercise, your risk of developing breast cancer also goes down. Again, observational as well as randomized clinical control studies. We know that when you start becoming obese, that the adipose tissue starts producing a number of pro-inflammatory factors. They start not just storing more fat, not just getting larger, but the adipocytes themselves start to change what they produce. So they'll start producing more of this TNF, more leptin, more retinol binding protein, lipocalin-2, interleukin-18, CCL2, which for this lecture is going to be really important. Not shown here, but also CCL3, chemokine ligand 5, as well as angiopoietin-like protein 2. Some of the other good factors, the anti-inflammatory factors, have decreased because of the switch that we've seen. We know that adiponectin, which is good for us to have lots of in the circulation, has decreased after we start gaining weight. And we know that SFRP5, or secreted fissile-related protein 5, is also reduced, and that's also a good beneficial factor that is anti-inflammatory that is being reduced as we start to gain weight. So the adipocytes are certainly changing. The circulating cells, the monocytes, are also changing whether we exercise or don't exercise. And again, I'll mention that when you start going back and you start looking at how to start studying, many of you had a lot of the elements of the diagram here in a different form on your midterm. You wrote it really well. I'll just ask you to be specific in terms of how you label things, and that's the only comment I wanted to make here. Some of you mentioned that there are changes in macrophages. But you know what? We have macrophages on both the left and the right. So indicating if it's an M1 or an M2 type macrophage is probably a better idea for the final exam if you're asked to label a diagram. Also indicating, for example, whether or not you have a T cell, if it's a Treg cell, etc., is probably a good idea. So just being aware of like how to label a diagram as shown here is probably a good idea. You'll get to see your exam in a few days anyway, so I'm hoping that you'll come and check out your short answers as well as your multiple choice test questions and get some feedback that way. Unless you don't want your midterm exam, it's up to you. If you don't want to see it, that's okay. If you want to see it, you'll be able to pick it up. There'll be an announcement available for all of you. But I also wanted to take a look at this. So yes, your adipose tissue itself may be impacted by exercise. You're going to be burning through some of those triglycerides that are stored there. And certainly they'll change shape, they'll change size. Some of the factors that are secreted as a result of that may change, but your muscle is also being very active during exercise as well. And we think that regardless of the whole factor related to the adipokines, are these myokines, the factors that are released from muscle as they contract. And again, there is one paper that's out there that says that you don't actually have to contract your muscles to benefit from exercise. All you have to do is imagine that you're contracting your muscles, and that's enough to cause the beneficial effects of myokine release. But we think that it's a biological phenomenon, that you have to keep doing this in order to have these secreted factors. And we know that one of the things that happens during the contraction of skeletal muscle, and these are the muscles that are in your biceps, your quadriceps, etc., the ones that are actively working during exercise. And one of the things that happens is that during exercise, we have an increase inside the myocyte, the muscle cell itself, of calcium. And this calcium trigger acts to turn on two different types of pathways. It can either turn on the P38 MAP kinase pathway, or it can turn on the calcineurin pathway. Both of them require calcium for them to function optimally. Now, there are two other pathways that get turned on. There is this Krebs pathway over here, the cyclic AMP response element binding protein pathway, or the Krebs pathway here, and that involves having Krebs phosphorylation, P300, which is a separate protein, as well as Krebs binding protein coming together. And when all three elements come together following the activation either by P38 MAP kinase or calcineurin, this becomes a transcriptional activator. You need all three in your muscle cells coming together after either P38 or calcineurin has been activated. And then what it does inside the muscle is every time it contracts, every time calcium comes in, this little complex gets activated, translocates into the nucleus, and causes the increased transcription, translation, and release from your muscles of interleukin-6. The other pathway that seems to do and activate exactly the same thing, also causing an increased transcription, translation, and release from the myocyte of interleukin-6 after muscle contraction, calcium entry, through a calcineurin or P38 MAP kinase induced pathway, is the transcriptional factor that comes together with AP1 as well as NFAT. And it works in exactly the same way, same types of transcriptional activation. Both of these cause an increased release of interleukin-6. So imagine every time your muscle is contracting, every time it's kind of at work, a little burst of interleukin-6 as a myokine is released from every single muscle fiber that is contracting. And we can actually measure this in your blood. And one of the interesting things that we see is that during exercise, that there is an exponential increase in interleukin-6 levels that are released into your circulation. Okay? So interleukin-6 is a factor that is actively released from your muscles every time they contract. And you'll see that when we start here at time 0, and the blue bar indicates that this is a 2-hour exercise regimen, that as soon as we start exercising, there's a slow buildup and then a massive buildup here, an exponential increase in interleukin-6 levels. And as soon as you stop exercising, look at what happens to interleukin-6. It doesn't stay high. It reduces almost back down to normal baseline levels within the first hour. So there is a benefit that we get from interleukin-6, but it doesn't stay there forever. It's a pulsatile benefit that we see. But the effects are also lingering in the sense that interleukin-6 will in turn turn on the activation of interleukin-1 receptor antagonist, or interleukin-1 RA receptor antagonist, as well as a delayed elevation of interleukin-10, which is the anti-inflammatory molecule. So yes, interleukin-6 goes up and down, but it acts as the first signal of other beneficial factors like interleukin-1 receptor antagonist, as well as interleukin-10. So just showing you this in a slightly different way. We're not really sure how this works, by the way. There are two different schools of thought. When your muscles start working, one thing we know for sure is that intracellular calcium goes up. Those factors, the p38 MAP kinase, calcineurin, both of them require calcium. We know that for sure. But the other possibility is that as your muscles start working, and this is why there's a delayed response, the other signal that might get this all to start working is a decrease in intracellular myocyte containing glycogen levels. So your myocytes have stored up glycogen, which is the storage form of glucose. And as it starts working and it starts chewing through glucose molecules, the decrease in glycogen might also be the trigger for this p38 MAP kinase or calcineurin induced pathway that eventually leads to interleukin-6 release. Now, I'm not going to go through this diagram in a lot of detail right now. I'm going to come back to this diagram a little bit later on. But one of the things that we know is that interleukin-6 has a number of beneficial effects on different organ systems. So one, we know that it increases lipolysis. It breaks down fat inside the adipose tissue. It has a beneficial effect on the liver itself, and it may or may not have a good effect on the brain. So a lot of people say that exercise is good for your cognitive skills and to help you to think, and interleukin-6 might be one of the ways in which we do this. For the time being, I'm going to skip through this diagram here on the right, so we won't have to worry about how it might activate GLUT4 translocation, although we've seen that before. So I'm going to tell you a little bit more about the effects of interleukin-6. When active skeletal muscle starts to contract, we increase the levels of interleukin-6, and then prolonged exercise of about 2 hours or so, we can increase the levels over baseline 100-fold. And we know that's a huge increase in the levels of a myokine. However, these levels return back to baseline very, very quickly. Within an hour, the interleukin-6 goes down very rapidly. However, we know that it goes up dramatically during this 2-hour period, and we also think that it's both calcium mediated as well as potentially glycogen content mediated as well. So you might think that's a terrible signal. This signal goes up after exercise but then comes back down really, really quickly. But the effect is actually longer lasting because interleukin-6 in turn turns on the interleukin-10, which is more prolonged. It stays elevated for longer periods of time, as well as increasing the levels of interleukin-1 receptor antagonist levels and increasing the levels of cortisol from the adrenal glands. So that's another factor that we have to take into account. So again, experimentally, how do we know that interleukin-6 is actually all that good for us? So one of the things that we know is that if we were to infuse in interleukin-6 into someone's bloodstream, that it would actually mimic the anti-inflammatory effects that we would see similar to exercise. So if we just inject in interleukin-6 into your bloodstream, and normally we would see interleukin-6 go up and then interleukin-10 and interleukin-1 receptor antagonist, we would see that we would get this elevation even if you didn't exercise. So we know that it has these anti-inflammatory effects. We also know that if you were infected with an endotoxin, so you have a bacterial infection that then produces an endotoxin, one of the things that we would be able to see is that the interleukin-6, if we were to give this to you, would actually suppress the TNF that's being released after you were infected or you had an endotoxin-mediated immune response. So all of these things together suggest that there is an anti-inflammatory effect that is being driven by this interleukin-6. So why might this be the case? So interleukin-1 alpha or the beta isoform normally binds to the interleukin-1 receptor, and when it dimerizes, like we've seen before, it will activate the NF-kappa B pathway that we've looked at, similar to the toll-like receptor system. And then one of the things that it does is it causes or produces inflammation in other tissue. So these are normally found on muscle tissue, on macrophages, as well as in the brain. So again, just keep in mind that where these things exist are important, and in muscle, it has a slightly different function. But in macrophages and in the brain, it usually causes the increased production of interleukin-1, more interleukin-6, TNF-alpha, all of which cause inflammation. And that's if this is normally binding. Now, after exercise, interleukin-6 has caused interleukin-1 receptor antagonist to be produced. An interleukin-1 receptor antagonist actually prevents dimerization. It prevents the activation of NF-kappa B. It prevents the inflammatory responses that we would normally see. And as a result, it is anti-inflammatory. And so we know that just injecting interleukin-6 will produce the same effects, and we know that interleukin-1 receptor antagonist has these anti-inflammatory effects just by blocking the interleukin-1 receptor system. So this seems to be one of the ways in which this interleukin-6 tends to work. It has this activation of this anti-inflammatory interleukin-10 and the downregulation of the immune responses that we see that would normally cause tissue damage. We also know that interleukin-10 as an anti-inflammatory agent is produced by these Treg cells. Now I asked you to make sure that you understood the function of these Treg cells earlier on. It was one of those earlier slides. What happens as you become obese? Treg cells go up or go down? So we are losing Treg cells. We are replacing them with other types of immune cells as we become more obese. These Treg cells themselves are releasing these anti-inflammatory cytokines that we've been taking a look at. So when you go back to that diagram that we had earlier on in the course, where you had on the left healthy tissue, increased production of the interleukin-10, one of the ways in which this happened was not just through the adipocytes, but also through those Treg cells that we saw earlier on. Now, I'm going to come back to these adhesion molecules and CD8, CD80 and CD86 later on, but we also blunt the immune response system as well. Now, this is a little counterintuitive. How many of you know elite athletes? Like, I'm talking elite athletes. How many of you know okay athletes? Not elite, but okay. So let me ask you this, without trying to get you to think one way or the other, or to bias your opinion. Compared to you, your friends who are better athletes than you, do they get sick more often or less often? A little bit of both. Sometimes they get sick, sometimes they don't. How about getting the cold? Do they get sick more often with the cold or less often with the cold? You don't hang out with your friends, I get it. It's okay. So it's a little counterintuitive, but because for elite athletes, they turn on this anti-inflammatory response so much, and because they're exercising so much, in fact, some elite athletes, including swimmers and long-distance marathon runners, tend to get more sick with upper respiratory tract infections. And we'll talk about this counterintuitive nature of this anti-inflammatory response a little bit later on. So again, these interleukin-10 is really important because it has a number of effects on both soluble released factors as well as downstream effector T cells being activated that might help to sustain an immune response, but we're turning all of this off. We know that in obese subjects that interleukin-10 levels tend to be lower, and that if we were to give obese individuals interleukin-10, it would actually help to reverse their insulin resistance. So interleukin-10 seems to be really important. Interleukin-10 gets turned on through interleukin-6, released from those myocytes, and we lose that as we start becoming more and more obese. And then again, interleukin-10 has a number of great effects, including insulin sensitivity, responds well to having insulin, make sure that your glucose levels in your blood are normal, and it also will protect skeletal muscle from obesity-associated macrophages coming in. Macrophages don't just go into fat tissue. Macrophages, by definition, will go into any tissue that they happen to suspect are having any type of immune reaction. So we'll talk a little bit more about that in the upcoming lecture. Here's something to think a little bit about though. I asked your colleague earlier on, how much exercise do you really need per week? 150 minutes, and that answer is actually 100% correct, or 150% correct, or 150 minutes. The reality is though, when I'm walking across Queen's Park and I'm slightly out of breath and I may or may not be sweating, and I'm doing that for about 20 to 30 minutes, if you were to look at me, I'm not exercising for that 2 hours. I don't have this huge spike in interleukin-6, but I'm still benefiting from that exercise. We don't have a good answer for why moderate exercise, which again, everyone agrees in the literature is good for you, like walking 30 minutes a day vigorously is good exercise. We all know that. That's a recommendation from Health Canada. But interleukin-6, nothing. There is no effect that we can see from interleukin-6. Now again, whether or not Irisen does anything, we'll talk about that a little bit later on, but short durations of low to moderate intensity exercise, none of this interleukin-6, therefore none of that interleukin-10, therefore none of that interleukin-1 receptor antagonist. This is well described. It is not a controversy. We just don't have a mechanism right now. It doesn't mean that we won't. Maybe one of you sitting there who are thinking about this, who are bothered by this, might come up with a brilliant mechanism to help explain why we don't get interleukin-6 levels being elevated. Now I'm going to come back and talk to you a little bit more about this in the upcoming 2 lectures. As we start looking more into exercise and then the controversies with exercise. Just out of curiosity, how many of you are taking M250 right now? A couple of you. So how many of you feel pretty confident with your immunology background? Yeah? Okay. Not bad. So I'm not going to go into a lot of detail about the different types of monocytes. Suffice it to say, we'll be talking about these monocytes, and these monocytes, again, on their cell surface have a number of toll-like receptors or TLRs. We know a couple of things. Exercise directly will cause an increase in interleukin-1, interleukin-6, which then again will have beneficial effects through a number of different mechanisms that we'll go into in more detail, including the production of interleukin-1 receptor antagonists and interleukin-10 through other cell types, as well as direct effects on monocytes. But the other thing that I mentioned to you is that myokines, through these muscle tissue contractions, will also increase. Sorry, I just lost my laser pointer. There we go. Will also increase the release of cortisol. All of you have heard of cortisol. Cortisol is a stress hormone, right? You've all heard that stress is bad for you, right? But in fact, you've also heard the opposite, that stress, when you're stressed out, you should go and exercise. But the reality is that exercise itself is a mild form of stress. It's not the psychological stress that leads to cortisol being released, but when you have acute release of cortisol from the adrenal glands, in particular from the cortex of the adrenal gland that sits right on top of your kidneys, it can also have some beneficial effects. It reduces tumor necrosis factor release or inhibits the tumor necrosis factor release from monocytes. And we know that actually now, and it's not science fiction, it is not something that is beyond our understanding in science. We know actually that there is indeed a mind-body connection. A lot of you have heard that the mind controls the body. We know that through this HPA axis and the sympathetic nervous system, that indeed you can actually alter, by being stressed out, your immune levels. And you can also affect your susceptibility to different types of diseases. So think about it this way. Most of you, after midterms and after final exams, are you going to be sick or not? You're probably going to be a little bit sick for a few days, mainly because you've been stressed out for such a long period of time that you've actually affected your immune system. And it's primarily through the cortisol-mediated factors that we'll talk about a little bit later on. And we'll come back to different parts of this lecture on Friday as well. So we know that the sympathetic nervous system can directly cause the release of different catecholamines like noradrenaline. We know that interleukin-6 can also cause the stimulation of the release of cortisol from the adrenal cortex. And we know that exercise can moderate all of these different types of effects. Now, why are we interested in this? We know that it takes time for interleukin-6 to ramp up. It takes about an hour or so after you started before you see significant increases in interleukin-6. However, we also know that these hormonal responses in things like cortisol and ACTH levels ramp up almost immediately as soon as you start exercising. And so we think that cortisol as well as adrenaline levels also are really important in this exercise-mediated, the good effects of exercise-mediated changes in your bloodstream. So catecholamines, for example, have been shown to downregulate cytokine production by immune cells. If you challenge immune cells with lipopolysaccharides, which are normally found on bacteria, these catecholamines will downregulate the immune-mediated responses. It's an anti-inflammatory effect, in other words. Okay, I'm going to probably just go through the last two slides really quickly and then come back to them on Friday. All of these things together seem to suggest that your cells are being affected by a number of things going on in exercise. Some anti-inflammatory effects for sure, but also some of the effects through noradrenaline as well as through cortisol. Some of these are going to directly impact these macrophages, and these macrophages are going to, in turn, also be affected by the types of secretions coming out of the different adipocytes, whether it's CCL2 or CCL3. And this gets back into these different types of macrophages, the M1 and M2 macrophages. Hopefully you haven't forgotten too much about your different macrophages. Exercise, we think, increases the amount of M2 macrophages, which increase the production of interleukin-10 as well as adiponectin. We think that these M1 macrophages, which produce a number of deleterious or pro-inflammatory cytokines, goes down, which is exactly what we want after exercise. Okay, and actually I think, just for today, because it looks like most of you are kind of thinking about Bio 220 anyways, I think I'm going to come back to some of the elements of this in terms of how these mechanisms might actually work to draw in different macrophages into the adipose tissue on Friday. So I'm going to start putting up these types of slides. These will be your summary slides that you should concentrate on while you get ready for your final exam. We're going to continue doing everything that we have been doing. Some of you made these suggestions about having these directly in your slides at the very end of the unit. So we're going to try to do it this way. If you don't like it, let me know, and we'll try to figure out another mechanism. Okay, good luck on your Bio 220 if you're writing tomorrow.